In the last class, we talked about phylogeny. How about we take a look today at the tool it uses the most, the one we always see around? Welcome students and Curious Minds Life. My name's Tarek, and today we're going to talk about cladistics. It's normal for us to think about the relationships between organisms. We organize family trees to see where we come from and to understand who we are and why we're like this. In science, it's no different, it's just on a bigger scale. Scientists, in the field we mentioned earlier, phylogeny, have a way of doing this with these family trees. They're super elaborate. But instead of just one organism, we have entire species. That's what we call cladistics. This science was created in 1950 by an entomologist named Willy Henning. However, it only started being used more frequently about 30 years ago. That happened because Willy was German and his writings were in German. So there was very little information available on the subject until everything was translated. This guy was so important that the entire theory proposed by Wallace and Darwin could only have a solid foundation because of the way he proposed it. For the kinship analyses that are done, identifying the oldest evolutionary patterns and helping phylogeny to work, these concepts are essential. But what is cladistics then? It proposes a large family tree where organisms are classified according to their evolutionary relationships and the time they are separated from their common ancestor. So, two species that came from a common ancestor should be connected. And the more distant the groups of species are from a common ancestor, the greater the distance between the groups. All of this, in a representative way, is called a cladogram, which organizes all this information into a tree. As we can see, the information placed within this diagram and the way it is constructed comes from phylogeny, and in particular, morphological and genetic characteristics. But how exactly do we manage to separate them and neatly arrange them within this tree? Some concepts in this regard are important, and we're going to introduce them to you. If we stop to think about it, all organisms at some point share a common ancestor. So how do we build a tree to group them closer together and organize them into smaller groups? For that, we use synapomorphic characters, which are those characteristics of organisms that are shared among them. And by no other, and phylogenetically, they are new. A great example of this is the hard wings that beetles have. This is a characteristic that only beetles have among all other insects. Therefore, it's a synapomorphic character of beetles. Another interesting thing is that we can say beetles belong to a clade, and if we extend that, insects also belong to a clade. So we can define a group with a common ancestor as a clade. Now, it can happen that a synapomorphic characteristic develops only in a clade, or taxon, that is extinct. In other words, a new characteristic. That only it has, but since it no longer exists because this organism is extinct, this is called automorphy. Another important concept is homologous characteristics, structures in an organism that share a common ancestor, which may or may not have the same function, but originated from the same place. For example, the arms of mammals, flippers in whales, wings in bats, legs in horses, and arms in humans. All of these came from the same ancestral structure and can be compared to identify the synapomorphies mentioned earlier. It's also important to mention in this example that when a homologous characteristic changes in a way that differs from the primitive structure in cladistics, we call it an apomorphy within a lineage, while the ancestral characteristic, which no longer exists in the clade, is called a plesiomerphy. So, if your cladogram is all neat and tidy, and all the organisms within a group share a common ancestor with clearly distinct synapomorphic characteristics, we have a monophyletic group. That is, a grouping that has only one common ancestor. That was the extent of Hennig's idea. A pure cladistics that represents organisms in the environment must necessarily follow the concepts mentioned earlier. However, since nature doesn't usually follow what we know, we find other ways to see how it behaves within the evolutionary processes we're familiar with. So we can organize organisms into other groupings, like polyphyletic groups, which are organisms that do not share a common ancestor. Instead, through convergent evolution, they acquire the same characteristic due to environmental pressure. Other groups are paraphyletic, which are sets where the organisms have a common ancestor, but one or more of them come from a different evolutionary origin. It's like the whole group is almost monophyletic, just with a few outsiders. But you might be wondering, why would I bother grouping organisms that don't share a common ancestor? Shouldn't we just stop creating polyphyletic and paraphyletic groups? In fact, the pure Cladistics originally proposed doesn't account for these extra groups, but they are very important for science. There are many reasons for this, but maybe the most important is that science isn't static. All the time, we discover new information that can change the way we understand a clade. So, the creation of these groups can help us understand and organize information in a way that fills in what we still don't know about the evolutionary history of organisms. And this is just a glimpse of what Cladistics can show you. There's still so much more to talk about in this amazing science. I hope you enjoyed the video. Don't forget to like and share, and remember that we have a new video every week. So subscribe to the channel and turn on the notification bell to always get the latest updates. Thank you so much.